this discussion on data science for biologists. I must say I'm excited that we have a privilege to have this in this session ongoing. I must say also that I'm impressed to see the caliber of people we have in contact. I believe you can all hear me loud and clear before I proceed. Can you hear me?
Yes, Mr. Smith, we can.
Can hear you.
Good.
Yes,
Okay.
sir.
Distinguished ladies and gentlemen, you're all welcome to Data Science for Biologists training program. In the next 6 weeks, we're going to have a very interesting discussion and training program where we expect to have outcome that will be beneficial to us with great impact in genomic revolution. We will look at next generation sequencing. We'll also discuss computational technology in various areas of research. It is important for us to note that this program is designed for those that have interest in core research. And we're talking about biomedical, biotechnology, agrobiology, and particularly those that have interest in biology-related research field. My name is Sani Hamza. I'm a fellow with the Medical Laboratory Science Council of Nigeria as a medical laboratory scientist. I'm currently a mentor and a facilitator for OmicsLogic Nigeria and Eureka, that is in partnership with Pine Biotech USA. And today we are to discuss and interact to get the best out of the content which we wish to put forth to us. For us to understand today's session very much well, I would like to take us through the sessions that we're going to be involved with. Now, this program is designed for students and faculty members with zero background in bioinformatics to readily catch up and begin using computational tools right on without having any formal training before. By the end of this program, we expect that researchers that have participated and students will go along thinking of how we can conduct and do our research work via the content we are going to be giving us to. And we are going to have 6 sessions. The first session is going to be taken care of today, where we discuss the NGS data and genomic sequencing analysis. Then we'll have a session on transcriptomics and introduction to metagenomics. We're also going to talk about multiple sequence alignments and phylogenetic construction. Then we'll talk about introduction to machine learning in biological data sets. And finally, by the 6th session, we'll talk about planning a bioinformatics project. So we hope with your interest and interaction as much as possible, if you go along with us, we should be able to put you through on how you can do your research clearly without any issues at the end of this particular program. This is possible because of a team of researchers and faculty members. So we have faculty and trainers that will interact with us during this program as follows. We have Dr. Harpreet Kaur. Dr. Harpreet is a certified OmicsLogic trainer and expert bioinformatician who will guide us on hands-on session and also assist the participant with projects and analysis during the program. Her area of specialization is in cancer genomics and machine learning. We also have Dr. Mohit Mazumdar. He's the OmicsLogic project mentor and an expert in big data computational analysis. Dr. Mazumdar has a PhD in computational biology and over 10 years experience working with industry and academia. But we are excited to let us know that we have, for this particular cohort to be organized, we're excited to let you know that the Nigerian team have been put forward to see that this works out well. And in our midst, we have also our distinguished Mr. Adekunle, who happens to be a faculty with Lee City University. He is the director of Eureka. We also have with us as also a mentor and as our community manager, Dr. Adetayo Aborishade, who will also mentor us and also attend to all our needs as expected. So this is the team that have been put forward to see that we have good content at the end of this discourse. Now going to business. Some of this omics science includes genomics. The human genome is over 3 billion base pairs long, with over 20,000 genes and many unexplored dark matter that is being explored. Learning about the impact of variants and all this content is important for us to take note of. So today, our main discourse will be on how we look at genomics, then later on X-ray next generation sequencing as much as possible. When we listen about genomics, what's the first thing that comes to our mind? Now, the first thing that comes to our mind when we talk about genomics is for us to look at how some basic things come into together. Now we talk about the codes, the gene, how they are passed from cell to cell. There are about 3.2 billion letters in total, encoded to about 22,000 proteins. Now it is important for us to note that genes are made of biochemical entity called the DNA, which is in short form called deoxyribonucleic acid. The DNA molecule is a double helix that is two long, thin strands twisted around each other like a spiral staircase. The DNA is the backbone of each strand, is made of sugar and a phosphate, and these two strands interact in such a way that through the nucleotide base. It's important for us to take note of that for us to be able to understand and advance in the content we need to get us to know. In humans, generally speaking, genes vary in size from a few hundred DNA base to more than 2 million base. An international research effort called the Human Genomic Project, which worked to determine the sequence of human genome and then identify the gene that contains that human DNA carries, estimated that we have about 20,000 to about 25,000 genes. It is key for us to note that the human gene lists, as we say, it is hard to overestimate the importance of the human gene list. Series of studies have gone further to understand this so that we can be able to identify disease-causing agents and understand cancer or variants as it happens. Some years back in 1990, a research work was taken over. The primary goal of the Human Genomic Project, the HGP, which lasted between 1990 to 2003, was to determine both the DNA sequence and the location as estimated with over 100,000 human genes. Now, scientists made efforts to do this research, understand how we can be able to lock up each genomic workup with identifying it with where it belongs to. Now, at the end of the day, what came up was that it's so massive that for us to be able to move further, we need to get to understand certain basics. Now, the basic includes that the DNA is made up of nucleotides. Now, this nucleotide we talk about, a nucleotide is made up of 3 components. The sugar called deoxyribose, the nitrogen containing a base, and the phosphate group. Now, the nucleotide, as it is, as basic as possible, are joined together to one another by a covalent bond. And that form between the sugar of one nucleotide and the phosphate of the next, resulting to an alternative sugar phosphate backbone. Nucleotides are of 2 complementary strands connected together are called base pairs. Distinguished ladies and gentlemen, these base pairs containing 2 strands of DNA, when referring to the number of base pairs containing in the DNA sequence, are called connected bases. Each strand of the DNA molecule consists of a sequence base that is complementary to the base in opposing strands. And as you can see, nucleic bases are of 4 types. We have the adenine, the guanine, thymine, and the cytosine. The basic thing we need to understand here is that the central dogma when it comes to this is to understand how we move from the DNA replication down to transcription, then translation. One question that should be bothering us in our mind is, with all this I have said so far, how are we able to push through? With the advancement in next generation sequencing, what it has brought to us over the past few years, according to the Moore's laws, which says that we can expect the speed and capacity of our computers to increase every couple of years, then we will pay less to do a full body sequencing. If you look at the cost of genome in 2003, it will cost you about $100 million. In 2017, it is as less as $1,000. That should tell us that with advancement in computer, it allows us to do a dry analysis, having data, having information, and doing our sequencing as much as possible to be able to have good interpretation to do the needful when it comes to dry research. One of the things that you should note that when it comes to next generation sequencing, it allows researchers to explore genetic variation like never before, down to a single nucleotide resolution. Whether you need to identify a specific variant from a focused set of region, or look more broadly to a potential causative variation, like a whole exome sequence or a whole genome sequencing, this is very possible. Another thing we should note that a whole exome sequence is targeted, and we can actually view the protein coding region of the genome. Now, this also allows us a reliable and sensitive detection of code variants. Now, with NGs, it is faster and cost effective. You can imagine in 2003, it will cost $100 million. In 2017, it's about $1,000, and we are good to go. Now, we need to understand the types of next generation sequencing we need to get. Basically, there are 3 types. We are looking at now as a targeted sequencing. Here, we can investigate these, that is a specific region from our sample, and completing targeted sequencing. The targeted sequencing provides us both high coverage and in-depth look at what's happening within the specific region. Now, such localized sequence might be useful if you wanted to identify mutation that you suspect lead to a particular disease such as cancer. Also, the target sequence works best between the 40 to 400 genes. This also allows us, it's ideal for looking closely to small specific region of genome. It provides high coverage of gene and allows us to see single base pairs variation, such as insertion and deletion. It also generates small amounts of data, potentially allow us for easy analysis than some of the larger scale sequencing techniques. Then we also have the whole exome sequencing. Now, the whole exome sequencing, in order for us to get this clearly, it's on the large scale. Here, we can run a whole exome sequence which focuses on the protein codes coding genes. And it gives the broadest view of the sequence. Now, one of the things we need to understand is that the major advantage of whole genome sequencing is that it allows us to use both coding gene sequencing and non-coding gene region, providing us with more information about the information within the genome. Now, let us understand about the different types of sequence reads based on length, size of sequence. Now, small read sequencing up to 250 to 300 base pairs up to now, but the base pair between 100 and 150 is still standard. The basic thing I want us to take note here is that when we have a hands-on session, we were able to explain clearly about the small read sequencing, the passable and the minion, the short reads, and likewise the long read, as clear as possible for us to understand and be able to apply this. Now, as regards for NGs, we should note that we have short read sequencers and long read sequencers. Now, in your wet lab, this allows you the opportunity for you to do the necessary thing you need to do, and the data are being transloaded for us to analyze and get good outcome. So in next generation sequencing, one of the important things we need to take home with is that when we move away from genomics and we move down to transcriptome sequence, is a major advance in the study of gene expression because it allows snapshot of the whole transcriptome rather than predetermined subject gene. The whole transcript sequence provides a comprehensive view of cellular transcriptomal profile at a given biological moment and greatly enhances the power of RNA discovery. Now, as we move along, we're going to explain to us how this is possible. Now, let us understand how this is allowed, how this set is put in place. The NGS technology captures the DNA sequence and generates the digital data using several steps. Here we have the whole genome DNA is isolated. Then it is shred and placed into a flow cell. Inside the flow cell, the DNA fragment amplified using the bridge PCR application. And the flow cell is then inserted into a sequencer. The sequencer is a machine-used image analysis to capture each letter in the flow cell fragment by analyzing visual pattern and converting them to sequence letter. That is the T, the C, G, and A as seen here. Eventually, it will produce reads typically between 30 to 300 base pairs. Under sequence, data is stored in several different file formats. We have the FASTQ, FASTA, GTF, the SAM, and the BAM. Now, the FASTQ file provides us with reads from the sequencer. It's a text-based file format that stores nucleotide sequence of reads and the quality of each letter. The FASTA is a text-based format for representing DNA and protein sequences. Typically, this is the reference file for the genome. Then we'll go to the GTF, that is the genome transfer formats. Then we'll talk about the SAM and the BAM. Quickly, let me just talk about the types of mutation so that we can be able to go to questions and answers and do the needful accordingly. Briefly. Now, usually, DNA is incredibly stable and allow genetic codes to be maintained and ultimately passed onto the upstream. Despite this stability, there are many instances where there are changes in the DNA. Now, these gene changes in DNA is referred to as mutation. And when this happens, the mutation are characterized by the change it produces within the DNA. And we have what we call the point mutation. Point mutation is as a result when there is an alteration at a single base pair of nucleotides. And there are three types of point mutation, such as we know, the substitution, like what we have, you can see here clearly, insertion, and deletion. In addition to point mutation that affects a single base of the bases in DNA, there are also mutations that can change the whole more than a sequence base, a single base. These can affect a whole section of the DNA, dramatically altering the gene expression and protein function. For example, DNA sequence that is insertion improperly or more times than normal results to insertion mutation. Then an inversion is as a result when a sequence of base reverse orientation within a DNA molecule. Another type of mutation is called translocation. Translocation can happen in two different ways, within the same chromosome and between two chromosomes. Now let us look at some mutations that can be encountered and the impact that they have on genome. We have the first one known as missense mutation. A genetic alteration in a single base pair substitution alters the genetic code in a way that produces the amino acid that is different from the usual amino acid at that position. Some missense variation or mutation will alter a function of the function of the protein, and it's also called missense variation. Then we also have what we call nonsense mutation. Now, the nonsense mutation also involves a single alteration of the DNA base pair. However, in the case of nonsense mutation, the single change results in the production of a stop codon, thereby terminating protein synthesis prematurely. As a result, here there is incomplete protein form that may function or may not. As such is known as nonsense mutation. And lastly, we have what we call the frame shift mutation. Now, this occurs when there's any addition or deletion, the mutation results in a change of the gene reading from the frame, which includes group of 3 base that is encoded for the amino acid. The change in the reading frame alters the group of the base and subsequently changes the amino acids that are encoded. As a result, the encoded protein becomes non-functional, and this is known as the frame shift mutation. Also, when we talk about codons and amino acids, a change even at a single base level can be enough to change the type of protein expressed. So let us see what happened. Each amino acid is represented in the DNA of a set of 3 base pairs, referred to as a codon, that is A, T, and C. A mutation in codon sequence can result to a change in the amino acid produced. The result in altering the structure and the function of the protein. If you look at this table clearly, you will see clearly here how a codon could impact the amino acid expressed. Those both insertion and deletion can change the DNA sequence and the RNA and the protein level by changing a particular codon. And that is clearly seen when a change occurs. Lastly, I want to just let us know that what is the significance of a DNA variation? So far, so good. When mutation happens, it plays a significant role, particularly in cancer development, or better we say, in the drive of tumor formation. As we all know, there are various important genes that play key roles in cell cycle and regulation of cell growth. Any disturbance in those genes will definitely cause a challenge in the outcome of the codon that exists. Now, it is clear for us to take note that as researchers, we need to first understand the DNA, understand how mutation occurs, then how the implication of this happening. Now, when it comes to NGs, the reality that comes to play is that it allows us to express ourselves and ensure that when analysis is done and you have data, you can be able to do the needful accordingly to see that the data you have gotten so far can be analyzed and put into perspective. In the next few minutes, we're going to entertain questions. I'll pause for now, and we're going to have a live session for us to understand how we need to apply what I've said so far. We have just talked about one of the omics today, and today we are talking about genomics. We're even going to talk about transcriptomics and metagenomics before we now talk about the machine language. So the next few minutes, I'm going to pause now and attend questions via the chat box. So you have your questions. We appreciate you put your questions in the chat box. While that is going on, I would like to introduce down to the platform to allow us to be able to do live on session with Dr. Adetayo Aborishade. Dr. Adetayo Aborishade is our community manager and also a mentor and facilitator. He's going to run through the genomic pipeline and explain to us how we are going to apply what we have learned so far in the real world, particularly running our pipelines and having hands-on what we have discussed so far. So in the next few seconds, we want us to put our questions on the chat box. Then we will call on Dr. Aborishade to come on board. Dr. Adetayo.
Thank you, Mr. Sonny.
You can share your screen now so we can go
okay.
to
Thank you, Mr. Sonny, and thank you, everyone. It's glad to have you here, and I hope you can all hear me.
Yes, very loud and clear.
Okay.
Yeah.
Anyway, I would love to start with the question from David Akul. He's asking how that can a poor DNA sequencing analysis lead to mutation? David, if I'm correct, that's your question. Hello, David. So maybe the best way to answer this question is maybe I should try and rephrase the question in a bit to make all of us understand. Now, even if the goal of next generation sequencing is to be able to sequence data from our samples, be it a tissue sample sampled from the blood or serum, or metagenomic sample, environmental samples. But NGS is not perfect, right? And that's why in a FASTQ format, there are quality scores attached to each call. It is not every A that could be A. Once NGS calls an A, it gives you a quality score that tells you how reliable is that score, right? So that the first thing you do when you have sequence data is to do what we call data preprocessing. And part of data preprocessing is such that you'll be able to trim away PCR duplicates and poor calls with low quality bases. So when this is done, your chances of actually getting a true mutation and not poor DNA sequencing will be low, right? So the first thing we do after sequencing data is to data preprocessing, which removes PCR duplicates, which removes adapters and trimmers, and which removes low quality bases. Once this is done, any variant you get, any mutation you call, has a high reliability. So the DNA sequencing itself will not cause mutation, but poor reads might make you presume there is a mutation. And there are robust bioinformatic algorithm tools to prevent this and minimize it to a small amount. So David, I'm sure you understand that. Thank you, Mr. Sonny, for a very comprehensive introduction to NGS and genomics. And for most of us, some of us are clinicians, medical lab scientists, molecular biologists, and molecular scientists. So we are familiar with most of these things. And the first question we should ask is, why Pine Biotech? What is the essence of this OmicsLogic data? Why are we doing this? What is our goal? How do we plan to make this community research-oriented and vast in bioinformatics? And the way Pine Biotech has done this is by creating a couple or a plethora of educational resources. Before I go on directly to the hands-on sessions, I want to introduce us to these tools that Pine Biotech has as a company to effectively communicate and convey to the basics, the knowledge of bioinformatics. Please go with me to explore the first tool, the so-called learn.omicslogic platform. Please let me know if you can see my screen. Can you see my screen? Hello, Mr. Sonny. Okay, thank you, Dr. Nancy. So this first data set is the learn learn.omicslogic.com. I'll put this on the chat forum so we can all have access to it. The learn, please use HTTPS learn.omicslogic.com. This is the first educational portal that Pine Biotech has. And in this portal, if you're coming, even if you're not going to join us for this program, you can still register for free and utilize all the available educational resources. If this is your first time on this platform, just click on register for free, where you'll be led to sign up. I'm already logged in, so I'll log out again so that I can do this ab initio. So click on register for free. If you're a new user, create an account, input your name, your email address, and your password, then click sign up, right? I'm already registered, so I'll just click sign in. And it takes us back to this platform, right? Please, how many of us are here? Or how many of us are having issues to come here? How many of us are here? So we can take it step by step. The learn.omicslogic.com. Go to home, register for free. If you're a new user, you create an account, enter your preferred name, your email address, and an appropriate password. Then click on sign up. Once that is done, you're back to this page. And the first thing you see in this page, aside my username, the points I've been able to accrue, my level. If I click on this, there are many things that I've done. You will see all the achievements, all the courses completed, all the lessons, all the steps, and all the code blocks that I've run, right? If I click on this, you can see the same thing. These are all the codes that I've run on this platform, right? But if you're not interested here, just click on the courses. Sumaya, most of all the courses are not free. There are premium courses on this platform. However, all the courses are free for registered participants. If you are registered on any of our programs, like this one we are starting today, you will have access to all these courses. But if not, there are a lot of free courses on this platform. So once you click on the courses, you see a breakdown of all the available courses, right? For most of them, there are courses on introduction to bioinformatics. You can either go to this way, or you can just search for a specific course. For example, today's topic is genomics. Let me type genomics and see. This is a course on genomics. It's the course 3 on this platform. And genomics has 8 lessons that will take an approximate 10 hours to complete, right? And you can see that I have completed genomics 9 times. As a clinician, genomics and transcriptomics is my forte. So I'm always coming back to genomics because on this platform, you always have new things to learn. And if you go down, you will see that the DNA structure and variant that Mr. Sonny educated us about there, not only is it head, you can see this one is learn. This is an asynchronous, well-paced theoretical background, right? And if you can see here, you can see that this is our code, meaning that on this platform, you also have access to learning coding via OWL, as you can see here. So these are codes, these are the theory, this is the code. So you do the theory and you do what? You learn the codes. If you're interested in other courses, for example, if you're interested in transcriptomics, you can log out, you can click transcriptomics. You see that the course 6 on this platform, which consists of 29 lessons. 29 lessons on transcriptomics, right? And if you go down, you see the breakdown. This, when you see learn, those are asynchronous self-paced courses that gives you the theoretical basis of a multi-omics model, right? You see all this learn. If you click load more, if you click on load more lessons will be loaded. I can see under transcriptomics, PCA, supervised machine learning, all these courses with these yellow diamond images, they are premium courses. Either you access a program by paying for a program, or you go to each of these individual courses and make payments for them. Payment ranges between $10 to $45, depending on the course you pick. But I can, as I said, I can assure you that you can see that most of all these courses also have free resources, right, that you can avail yourself of. So this is our first platform that I would love to introduce us to. For new users, kindly just go on, like we said earlier, register yourself and avail yourself of this. The other platform, which Pine Biotech also has, is known as the server tbio.info. I would also love to put this on the chat forum so we can see it. The server.tbio.info, right? Now, this is the main platform where we run all the bioinformatics model, bioinformatics analysis. And today, I run some of them. If you have an overview of this platform, you can see that there are different submodules depending on what data you want to analyze. This is for NGS data, and underneath it, you can see transcriptomics, genomics, DNA. You also have mass spectroscopy data. You also have the data integration and modeling, right? Where here you can see you can do core, robust, advanced data analysis, supervised machine learning, unsupervised machine learning, factor analysis, and differential expression. All this can be done either with your own personal data or with sample data sets that have been integrated onto this system, right? Today, we'll see some of those demo data sets. For example, if you follow me and click this, you can see all these demo. These are sample data sets that have been imputed on this platform, right? Server.tbio.info is an artificial intelligence, smart, cloud-based server where at the touch of a button, you can perform bioinformatic analysis, right? You don't need to upload because molecular biology data is big data. It is large in volume. You can do analysis running into 45 gigabytes and much more, 200 gigabytes. And this might either stress your system or you might need to actually utilize a supercomputer. And in a resource-oriented setting like ours, how many universities have supercomputers? How many labs have supercomputers, right? PyBiotech has maneuvered around this circumspectly by the production of this platform, which is cloud-based, meaning you get your data and just load it on the cloud. And this platform will run it for you, right? That's the second platform. So are we here too? Are we following along? Any questions? Any questions about the platforms? Yes, David. Not only can you work with R, right? You can also work with Python. There's a third server that's also another platform, which has somehow been integrated in that first one. But if your goal is just to run codes, then this site is for you, the code.omicslogic.com, which is our third site, right? Let me first make it clear that we are not planning to make you hardcore coders. We are just teaching you step-by-step coding that is relevant for bioinformatics. I also put this on the chat forum, the code.omicslogic.com, which has been integrated into the learn.omicslogic.com. But if you still want to do coding, and which that is essentially what you want to do, you can still make use of this website. It's code.omicslogic.com, right? I'm also inputting it on the chat forum. Code.omicslogic.com. Also here, you can just come here, get started, create an account like earlier, and continue using it. There are a lot of free courses, right? But it also has some premium courses where you probably also need access to. This is opening, and you see, just like the other platform, you have things to do. But here, not only will you work on R, but for some of them, you also work on Python. So to answer David, yes, you also have some Python codes that are both on the platform and this, right? So these are the three websites, three educational platforms where you can go and learn each of the multi-omics: genomics, transcriptomics, metagenomics, and also advanced data analysis, machine learning, and use this analysis to visualize your data, to understand your data, to predict patterns from your data, and to interpret cogent patterns from your data. And lastly, we also have projects that have been projects and studies that are part and parcel of this learn.omics.com, where you can learn how some authors, how they've done analysis, how they've analyzed and gotten reasonable meaning from their data. For example, you can see this is a project which is TCGA Liver Cancer Project, right? You can see here, this is a step-by-step analysis of this publication, how the authors analyzed each step before it became a published data and is domiciled in NCBI. Not only this, you also have other projects. This is a project involving COVID-19 and the pathogenesis of SARS-CoV-2, right? These are almost 6 lessons. You can go through each of them and learn how you can make use of this multi-omics data in arriving at a sensible, circumspect, and clinically relevant outcomes from your data, right? So all these and many more can be found on all our platforms. And all these and many more will also be taught throughout the duration of this program. Starting from today, which is the first session, it's going to be a 6 weeks, once weekly program, right? To cut short on the theories, to further buttress what Mr. Sonny has talked about. Let's, in the next few minutes, reason or learn how do we visualize genomics data. For clinicians, you have a patient, they are managing either for an infectious disease or for a cancer. And because of availability of NGS, you have been able to sequence either from the sample, the tissue sample, or from blood fluids like serum, blood, or other available tissue. And now, what do you first do? What do you do to this data? For medical lab scientists, you have done with the wet lab. You have extracted the DNA. You have done all that should be done. What else can you do to your samples? After PCR and all, what's next to be done? Right? This is the dry lab aspect of analysis. And the first thing we'll do is what we call, how do you visualize your genomic data, right? As I said earlier, there are requisite demo data sets, which I would love to make use of them, right? So for people to have access, please, just kindly go with me to this server.bio.info and let me know when you are there. I want us to go through it together so that you can, this program is a practical hands-on. We'll do it step by step together. If I'm too fast, you can call me back, right? Please, once again, server.t the information is, just click on it and let me know when you are there, right? If you are there, you can just say yes or put one in the chat forum. I would take some few seconds to wait for you. Thank you, Ajoke. Thank you, Cynthia. Thank you. Thank you very much. Mr. Sonny, please, if you can, for people that don't have access, there's a demo access which you can use to just follow along. Mr. Sonny, can you kindly help me put the demo access on the platform, or give me a second, let me impute a demo access so that every one of us here can have access to it. Thank you, everyone. Thank you, Zudin. Thank you, Ben. Okay. So this is a demo access you can use. You can use that username. As you come to the access, you can use this username, test@pinebiotech.com, and use that password. Thank you, Olabilekon. That will give you access to this, right? So because what we are working on is genomics, and is what is genomics data. So we'll scroll down to this genomics aspect, right? And if we click on this arrow beside mutation variants, we'll have these 3 demo data sets that we can make use of for today's visualization. I would love to click on this variant calling. These are for cell lines. If time permits, I will also try and visualize this SARS-CoV-2 if possible. But let's start with this first. So once we click on this, right, we are faced with this plethora of tools, plethora of bioinformatic tools, right? And these tools will allow us to do any analysis whatsoever on our genomic data. But remember, the first thing I said was that before you run a data analysis, there are various steps, right? And these steps are also highlighted in this server. If you look at this place, if I magnify this a bit, the first thing you see is what? Data preprocessing. We just don't want, we know that NGS itself is not 100% accurate. We could have some poor data from poor reads, right? In the process of NGS, we've made use of adapters. We've made use of PCR duplicates. And we need to remove all this so that our data can be reliable when we begin to call variants, right? So the first step here is what is known as data preprocessing, and where we can use any of these tools. We can use PCR clean to clean PCR duplicates. We can use Trimomatic to trim adapter sequences and also trim poor reads, right? That's the first step we do. Most times when you have your data, this is the first step. But in this case, because this our data has been cleaned before incorporation into this website, into this platform, we're not going to do processing today. The next step, which is the first step we're going to do, is when you take samples for analysis, how do you know which part of the genome? Mr. Sonny said our genome is 3.2 billion nucleotides. When we run our reads, how do we know exactly where these reads are coming from? Is it from chromosome 1? Is it from chromosome 22? Or is it from sex chromosomes X and Y? We first need to be able to align them first before we can begin to read any meanings into either there's a mutation here, there is a point mutation there, or there's a variation otherwise, right? So after data preprocessing, the next step is what we call mapping or alignment, right? Where we align the result of our reads to a control known as the reference genome, right? And that's what we do. And these algorithms, STAR, BWA, and BOTI2, will give us the chance to do this. Meaning, Once I click start, meaning I want to start this alignment, this process, I'll click OK. You can see two things are happening. I said our server is smart and AI enabled. The first thing you can see is that some cells become what? They become yellow. Meaning the platform is telling me my next option is to preprocess with this or map with this. As I said earlier, this is a demo data set and preprocess has already been done. I'm just interested in what? In mapping our reads to the reference genome. So I'll make use of Botai2 to do that. Once I click Botai2, this will explain what Botai2 does. And if you read from here, is for what? Aligning sequence reads to large reference genomes, right? And it is designed to work with a range of read lengths from 50 up to thousands. So this is what this is going to do for me. Now I've clicked Botai2, right? If you check next, something else is already, visualization is what? Is already being highlighted, meaning visualize my genomic read, right? And this also, there are various genome browsers we can use for this. We have JBrowse, we have IGV, and we have UCSC. But the JBrowse, the genome browser, which has been fully integrated into this platform. So I'll click OK, that all I want to do is visualize. If you check this, you say that the next item to be highlighted is to end my pipeline, right? Which I'll also go ahead and click on end. This pipeline is complete. How many people have been able to follow with me this far? For David, we are working on human genome, and the human genome 38 version is imputed on this T server. So this is HG38 that we're working on, the 38 build is the reference genome. Reference genome for each species means genome for that species, right? The human reference genome project, which got a lot of money and took almost between 11 to 13 years, was completed in 2001. So now we have a reference human genome where we can use as control for all reads from humans. And not only that, plants, some animals and mammals also have their reference genome. Meaning once you take any read from this species, you can align into that reference genome and to tell you the location, position, chromosome of your reads, right? Miriam, please just copy and paste. It is very case sensitive. Please try it again, just copy and paste, right? Have you been able to follow me to this point? Please, if you have, just type 1 or put yes, so that we'll know, or if I have to take a step back a bit so that others can follow along. Thank you, Dr. Adetunji. Thank you, Elvis. Thank you, Mujib. Thank you, Zudin. Felix, where exactly was the problem? Thank you, Dr. Tuga. Zainab, you came late because of Felix and not because of you. Just a step back. Dr. Tuga, you're saying both. In the next few seconds, let me just repeat this cycle, right? I'm saying in this case, what we want to do is analyze NGS data, right? So we had NGS data. For now, you can also make use of your own individual data set. But for today, for this practice, I want to make use of a sample data set that is domiciled in this platform, right? And if you click this, you see 3 demo data sets, right? You have variant calling lines, you have SARS-CoV-2, that's the COVID-19 virus, and for Ebola. So I'm saying I want to make use of this pipeline for a cell line, right? Once I click on it, it brings me to this plethora of tools and shows me the various processes depending on what you want to do, right? And I'm saying for all data, the first thing to do is what we call data preprocessing, where you clean your data to remove poor quality bases, PCR duplicates, adapter sequences, so that whatever result you get downstream can be reliable, right? So and I said, on this platform, you have all this to do that. After preprocessing, the next thing to do is to align your data, your sequencing data, to a reference genome. If this data is from humans, you align it to the human reference genome. If it was from rats or mouse, you align it to the reference genome of Mus musculus. If it's from other animals, you also align it, right? What that does is that it shows you exactly where your weights map to, right? And I said the tools for this is STAR, BWA, and Botai2, right? And I said for today's, what we just want to do today is visualize our genomic data. We have done wet lab, we have sequencing reads. Let's visualize it, right? So I said once you click start, meaning you want to start a pipeline, you click OK. Because this is already a preprocessed data set, I don't want to use PCR clean, right? What I want to do is just map to the reference genome. So I'll click on Botai2, right? Botai2, what you can see here, it says Botai2 is an algorithm for aligning sequence reads to large reference genomes. I click on this. Now you can see every step of the way, the platform is telling me what to do next. It's giving me options, alternatives. But here it has only given me one alternative, which is to visualize. So I click on visualization. It also gives me a small info, just small info about what I should expect, right? I click on OK, right? When I'm done, it's telling the next step, which is to end this pipeline. That if my goal is to visualize, I'm done with this pipeline and I end, right? When I end, I click OK. When I click OK, this is my result. The pipeline I just ran, that I did Botai2 to map, I visualize and end, this is the result. The result is in this output file, which is demo pipeline output file. And if you look at it carefully, you can see this button, this blue button that is showing visualization, right? If I go ahead and click, as you can see, what is happening? It has opened one of our genome browsers known as JBrowse. Can you see? JBrowse is a genome browser, and that is the preferred genome browser for server.tbio.info, right? Let me magnify this so we can see it clearly. Let me also minimize it, right? So what we can see here is these are the samples. We have group 1 and group 2, right? We also have the reference sequence. So let me kindly click on one of the samples so that we can see. So this is sample 1. And you can see all these color changes. This is the samples that I've read. This is these samples being aligned to the reference genome. Let me minimize so that we can see it clearly. I think it's okay here. Okay. So the first thing you observe is if you keep running, if you keep moving this around, you see these red and blue lines, right? You see some empty spaces in between. These red and blue lines, these are our sequences. These are our sequences, right? And if you look at this, these numbers mean positions on the reference genome. meaning at position 100,000, you have these sequences. At position 150, 200, you have these sequences. If I minimize more, you'll be able to see them clearly. Then if I want to go to any position, for example, let's assume I want to go to position 50,000. I'll just click on it. It will take a while to load. No, Zainab, you don't need JBrowse pre-installed, right? You to just go. If you're coming, you can either open JBrowse directly by yourself, or server.tbio.info will open it directly for you. We open it directly for you automatically because it is integrated with the platform. Dr. Adaja, I'm not neglecting variant calling, right? Variant calling is also one of the things we do under genomics. And if you're part of the last cohort, we have gone to variant calling retrogressively and comprehensively, right? Today, we're just trying to show how to visualize data. Variant calling is another aspect where we'll go to if time permits us. So what we have seen here, these peaks are showing the coverage of our reads at these places. And at this point, I begin to see the sequences. We're beginning to see this. If you keep magnifying this, you can see that the position is already written here as chromosome 1. So what we are doing now is visualizing chromosome 1. If you click on this, we can visualize any aspect of the chromosome, even chromosome X and Y. But presently, we are visualizing what? Chromosome 1. And what position on chromosome 1? We are visualizing 4829, this to this, which is 21.26 kb. That's what we are visualizing presently, right? And let's just focus on a segment, right? If you focus on this is a sequence that has been aligned to the reference genome, right? And if you want to see the reference sequence, let's keep zooming. You see, these are the individuals' GTA sequences. This is the reference genome, right? So what does this mean? These colors, what do they mean individually? What knowledge can we gain from this, right? Once you see a red color, it means this is what? A positive strand. It is a what? A forward strand. You know, DNA come in pairs, the forward and reverse strand. So this red means this is a forward strand. Why this blue means is a reverse strand, right? If you click on, to get more information about this, you click on it and opens this, right? It tells us the sample, the total quality score, the position, and the strand, whether positive or negative. Then it gives us this each sequence and the quality call of each sequence, right? We can also see the length of this. This is a 101 base pair sample, just 101, right? All this information are available, right? This, if the only thing you need is the read of this sample, this is the read. This is the fast A read of this sample. It's just AATTCCC, and so on and so on. Let's click on a blue sample to understand that. If you see a blue sample, you can see that the first thing is it is negative. Meaning what? It is a reverse strand, right? The sequence and quality of which is also shown, right? JBrowse gives us a lot of information. If you can see that even though these are both shades of red, this is lighter, right? It's telling us that this lighter sample it is not properly paired, or the qualities are not as high as this, right? Even for blue, you can see that this is blue. This is almost faint blue. All these are more, all this information can be gotten from this, right? And also, you can also see this is the total coverage of this segment. You can see this is very high, right? And if you click on this, it will tell you that a total of 571 reads, right, align to this reference genome, right? And this information can go a long way in making us visualize our sample. So if I'm looking at this sample, I know that it has aligned to this segment, but there is zero alignment to this segment. Please, are there any questions? Are we following through? If you don't understand anything, please kindly put it in the chat form, right? So this is how to visualize our data. The red for positive strand, the blue for negative strand. I can see, if you compare alignment here and alignment here, you can start, this is very low, right? So for David, if I'm trying to do variant calling in this region, I will be very circumspect because the coverage here is very low. But if you come to this place, you can see that the coverage here is high enough and sufficient to be able to call a variant or a mutation. But here you can see that here is very low. Here is also very low, right? So these are factors you put into consideration. Olamilekon, he said you need the reference genome. The reference genome is incorporated in JBrowse. And you can see that this sequence. Let me zoom in so that you can see. This sequence here, this first sequence is the reference genome, right? And anywhere there is variation will be shown. This here, you can see reference sequence. This is the reference genome. This is only showing our sample that's been aligned to the reference genome, right? So Dr. Tumba, what quality score of a codon makes it a sequence reliable? From Emmanuel, usually this is the, how do I put it? This is the second cohort. So most of these topics have been dissected in our first cohort. If you send us a mail at african@pan.bao, we'll try and link you up with resources and probably videos, session videos that will make you understand effectively what quality scores are, right? What are Fred quality scores? Right? It is not session, right? But send us a mail at african@pan.bao, and I assure you, we'll give you enough resources that will make you understand what exactly Fred quality scores are, right? Please do. You can, if you also go to Genomics 1 on learn.omicslogic.com, which is free, right? It will also give you more insight into Fred quality scores and quality scores of sequencing data. Thank you very much. So any more questions? Any more questions? Felix, go to learn.omicslogic.com, right? Go to Genomics, Introduction to Genomics or Genomics 1, which is absolutely free. This will be, you will learn more about these quality scores and how to either trim them away before you start your downstream analysis or how to include them in your downstream analysis. Thank you very much. If there are no questions, yes, David, you can. Let me click on this. Maybe it's not available. If once, David, you know, especially when we come to our coding platform, right? Once you have this sequence, you can go to how and use bow strings to calculate GC percentage. You can calculate the GC percentage of the whole sample or of different windows. Maybe this is percentage of 100 bases, of 200 bases, or all total your samples, right? The calculation is not automatically done in JBrowse. JBrowse is basically to just visualize, see where the changes are, see the coverage. For example, you can see that this place has a high coverage, while some places have low coverages, right? That's essentially what JBrowse is for. But if you want to know the GC percentage, that's also possible, right? And you can do that either on how, which if you go ahead to Genome 1 or Genome 2 and the how coding, you'll be able to learn how to check for GC content of a sample, either of the total sample or segments of that sample. But we all know the function of GC content, and it is possible. So thank you very much, David. Our time is fast spent, so I will not be able to show us, or we won't be able to visualize the other aspect of it, which is SARS-CoV-2. But as Dr. Raja mentioned, what is the essence of visualization? If you're not going to call a variant, right? Can we do variant calling via our platform? And the answer is definitely yes, right? And in the next 5 minutes or very few minutes, I'll quickly run and show us how to call variants or the so-called mutation variant calling on this platform, right? Here is also, we also use one of these data sets. As you can see, the title of this is what? Mutation variant. Parallel analysis of mutation variants. What do we mean by that? As Mr. Sonny rightly said, even though replication is almost perfect, it is still imperfect. And when the DNA is replicating itself, it makes a mistake of one base in every 1,000 replications. 1 in 1,000 bases, there is a mistake, right? And these mistakes can be point or can affect a sizable number, right? And it has been proven that some susceptibility to infections, some cancers, some differences in genotype and phenotype are due to these mutations, are due to these variations. So for every sample we have, your goal might be to know whether the cause of this disease, whether the cause of this cancer, whether the cause of this susceptibility is as a result of any of this mutation, whether single nucleotide polymorphisms or mutation affecting large segments of the genome. So let's also use this variant calling, right? As I said earlier, if you have your own data set, you can use it, but for the purpose of this demonstration, we'll do this, right? We'll click start as usual. You can clean if you want to. In this case, because we are doing something more robust, so let's clean. After cleaning, what do? We'll do bow tie. meaning we'll map to, and you can see the next thing that is shown is differential variant calling. It's taking us to this bioinformatic tool known as Strecker, right? Strecker is a tool that is used to call for variations in sample widths. And we'll click on Strecker, right? After Strecker, it tells us what it's going to do. Then we'll click on OK. Then if you want to use Somatic Sniper, you can, right? Maybe I should use Somatic Sniper in this so to fully explain it to us. Then I click on Somatic Sniper. Then I click on End. This is a typical pipeline to call variants. You start PCR clean for preprocessing, both Title for mapping, Strecker and Somatic Sniper for your differential variant calling, and you end, right? As usual, your result will be here, right? And if you want to see your results, you have to click this output. So let me click this output and download it. How many people are also able to follow along? Elvis, can you hear me now? Okay. Elvis, please check your audio. I think the others can hear me, right? So if you're able to follow along so far, just click yes or one. Okay, thank you, Elvis. Maybe it was a network problem. Thank you, everyone. Thank you, Cynthia. Thank you, David. So once you click on this, you realize that it has downloaded. So let me just go to my downloads and access. Okay. So this is the output file. And if I open it, you can see, right? Now, variant calls are formatted in a file format known as VCF, variant calling format, right? And this can only be viewed with resources such as IGV, right? But when you use Somatic Sniper, it gives you results in a table-like format. You can say this is an Excel format. Let me open this, and we can glean more information from this. So if you are there, please, once you open it up, you have this .xlx, which you can open in Excel. You also have this, which will open later in IGV. But for now, let's open this. Let's see what information we can glean from this before we come back here, right? Can we still see my screen? Okay. Please, the video will be made available. You can watch the video later and follow on the platform, right? Thank you very much. So here, you can see this information. The first thing you can see is the chromosome is shown, right? And all these are in chromosome 1. And if you keep going, you see that this will actually show you individual chromosomes, right? That's the first information you get. That's the first line. You see individual chromosomes. Then on each chromosome, you see the positions. Now, when you call a variant, it's not going to show you every aspect of the genome. That's not its job, right? It's going to show you the aspect of the genome where there are variations, right? It will show you chromosome 1 to chromosome 20. If there's a variation on chromosome 1, it will show. If there's a variation on chromosome 11, it will show, right? So don't expect to see all the genome. We are visualized with JBrowse. What we are doing is what we call variant calling. We want to see where variations are. So this is chromosome. These are the positions where these are variations. And this is what is very interesting, which I want you to see. That the reference based on the genome is what is available here. Let me click on this. Now here, you're expecting to see what? A C. right? And when we did our sampling, our sequencing reads, we have two samples, one sample for the tumor, one sample for the normal, right? Please, I hope you can all see. Please, if you can't see, please let me know. I hope it's large enough so we can all see. So on the normal sample, you can see that the reference is still what? Is still C, meaning there has been no change in normal tissue sample. But on the tumor, you can see that what is written here is M, right? If you are familiar with IU pack of codes, we know that not only do we have ACGT, we also have others. Like M means this is an amino base, right? If you also send us a mail, or for the people that are registered participants, we'll send you the IU pack of codes. You know what M means. Are we together? So, but one thing is sure, that in this tumor, what you have is not what? Is not C. M actually means either A or C, right? But in the normal, it is definitely C. But for the variant, it can actually, it could have been an A, right? And the reason I said so is, if you now check the somatic score, which is what Somatic Sniper will do for you, the higher this score, the more likely it is that this is a mutation, a true mutation, and that it could also have caused or have led to the carcinogenesis in this case, right? For example, this score is high enough to say, oh, this could be a true somatic score, right? Just by looking at these first 5 fields, you can give a lot of information, right? For example, let's look at another one. Let's look at number 10. In number 10, the reference base is C. Normal is C, right? But tumor is M. Here, the reference base is G. Normal is G, but the tumor is K, right? In K, K means the ketone, right? Meaning it can be a G or what? Or a T. But if you look at here, you see that this somatic score is what? Is very low. So it's likely that this is not really a true mutation. Even if it was a true mutation, it might not have been the cause for this mutagenesis, right? No, Zainab, the reference base and the normal are not always the same. There could be variations, right? There could be variations. These are things we'll discuss fully later. The time is fast spent. At this point, I would just like to ask for more questions, and I need to invite the program director to wrap up. If you have more questions, you can send us questions at africa@pound.bao, right? I'm putting it segment again. For the registered participants, please send your questions on our WhatsApp forum, on our Slack forum, and all your mentors, both local and international, will deal with it. If you still want to be part of this program, to learn more about genomics, transcriptomics, please, you can still join, right? This was made free so that you can be encouraged to join as part of this program. If there are no further questions, I would like to hand over at this point to Mr. Adekunle, who will end up the sessions. As I said earlier, please, you can still drop your questions. You can send us email at africa@pine.bao, and you can also call us, right, to get more information about this program. Thank you very much. Mr. Adekunle, over to you. Mr. Adekunle.
Good evening, everyone.
Okay.
Yes, please. Good evening. Please, I'd like you to make me a co-host so I can share my screen. Good evening, everyone, and it's been a wonderful time so far. Thank you, Dr. Aditayo, for that very elucidating section. Now, quickly, I'd like to just share my screen and take us quickly through few information sections to guide us moving forward in this program. Please, I hope you can hear me clearly.
Yes, we can. Yes, we can.
Okay, great. Thank you very much. All right. So, yes, we began this journey, data science for biologists, and in the next 6 weeks, we will be working together to learn about how to process and analyze high-throughput data sets, understand the logic behind them, and ultimately, one of the key sections I look forward to on this particular training is the project, develop your own project section that will be taken so that every participant can begin to think around using, key sets and bioinformatics key sets to analyze this data set in their own field of endeavor. Okay, so already, as we may be aware, Dr. Aditayo has shared with us a number of training materials and resources that we have. We spoke extensively about the server.p platform and as well as the code OmicsLogic.com platform, which we'll be utilizing in this training. As part of my duty this evening, I want to encourage and request that people who are yet participants who are yet join this particular program to um, come on board to join us because the program is still open for registration up until Wednesday next week. Because next session next week, which is on Wednesday, session 2, will not be available for all participants like today. So if you want to enjoy other sections of the program, you need to sign up. And it's still remains $25. You have certificate of participation, which is available to everyone that meets the minimum requirements for getting certificate of participation. And of course, there will be certificate of excellence equally for those who reach the 10,000 point milestone that is allowable for that. Quickly, these are some of the certificates from certificates of excellence earned by some of the participants of our past trainings. And I know it is very much possible. That is 10,000 points. And I look forward to seeing many of us on this cohort also attaining that. Equally, we have, this is the certificate of participation as an example for us to see. I'd like to move on to say that our goal for this training remains that we want to build a community of researchers. So like previous programs, organized, globally in other centers where we have participants going on to run and execute their own researches using, bioinformatic tools and equally presenting before their, colleagues in sections like conferences and workshops, just like what we see here on the screen. So we look forward to achieving that. As a matter of fact, the line towards the end of the year, there will be an opportunity for participants from our various previous programs to present their works in a conference section. So we look forward, that is the goal. So we are not here just to just to talk about these things or to take more shared resources and platforms, mentoring guide. Many of the questions that were being asked during the live sessions today, we still have to continue on our Slack page, via interaction. So comment questions should be posted on that platform. And mentors, any of the mentors, both locally here in Nigeria, as well as our international mentors, will be quick to respond to your questions and give you the necessary guidance. So these are the things we look forward to. And just to remind us, during this program, section 2 will be focusing on transcriptomics. I'd like to remind us that it's a section that will look at the various, aspects of gene expressions and the quantification of these expression levels. So I encourage us all to join that session next week. Also, session 3 will be on metagenomics. We already have the flyers, but these are some of the things we look into. We look at how microbial communities and live and function together, affecting a plethora of biological science fields like agriculture, health, the environment, and the rest. During that section, we will equally move on to take a look at multiple sequence alignment and phylogenetic analysis. That will be during section 4. And in section 5, we'll be taking machine learning. And so these are all meant to serve as introductory, pieces to guide participants on this journey into bioinformatics. And section 6, like I mentioned earlier, will be on planning a bioinformatics project. So I look forward to having us all, come on board and join the program. And importantly, I'd like to talk now about the assignments next week. Next section, which will be coming up next week, Wednesday. We expect participants to register and complete their profile on the learn.OmicsLogic platform. Of course, after registering the T-Bio server today, we're given a demo access and login details. I can very much assure you that has a lot of limitations. It is just meant for the demo sessions, but for every participant of the program, you'll be getting an email with a full access to the entire server.tbio.info platform. So that is one of the reasons you should register, so that the data sets we'll be using during the training, you can equally analyze them, and take advantage of that platform. So register and complete your profile, part of which will be us sending you the access, your personal login access to that server, as well as other resources. I also want to encourage every of the participants to go on and complete the courses on the learn.OmicsLogic platform. Ensure before next session you have completed the introduction to bioinformatics. We have some participants from previous programs also here in this cohort. I encourage at your spare time, you should equally look at them, because there are some variations, and some additions to those courses. I will share the links for these. The title for the introduction to bioinformatics is Bytes and Molecules. So it is not, you won't see it as introduction to bioinformatics on that platform. Equally, I want us to look at the Genomics course and also lay our hands on the transcriptomics. I think the Genomics has about 6 courses, packaged together in that particular course, lessons packaged together in that course. Also, let us complete the transcriptomics course. We'll be looking at transcriptomics next session, so it will be very much at an advantage to have taken the transcriptomics course, completed it, so that you move at a very fast pace, understanding and getting all of the things that will be shared during the live sessions. I equally want to encourage all participants during these next 6 weeks to begin to work on the project 1 and project 2, which are series of projects that have been taken from scratch, from the very beginning to the very end, analyzing and looking at COVID-19, for example, the origin and pathogenesis, how it was sequenced, and It's, one can identify the various variants. Equally on project 2 is Ebola virus. And here, for those of us who are very much interested in mutations, this will provide us with good understanding of all that has been discussed today. And of course, the assignment is for us to run demo pipelines as much as possible, while taking those courses. So without ado, the other thing I would like to mention is just to introduce us to the various social media platforms, but we can still always do much of this moving down the line. We have a LinkedIn page and a LinkedIn group, which I encourage every participant to join so as to be getting all of the informations and industry opportunities, openings, and the rest that is available, scholarships, and all of the details regarding the bioinformatics of big data analysis field. So join our LinkedIn group. We equally have our Facebook page where you can connect with us. And also for Jureca, which is the OmicsLogic Africa team, we have our handles both on Twitter, Facebook, and Instagram, where you can follow us and interact with us. I'd like to encourage us all to come along and join all of these various social media platforms. So on that note, I'd like to end this session today. Time is fast spent. We want to appreciate everyone who made it to this point with us and look forward to interacting further with you and engaging with you down the line. So if there are any questions, please quickly you can ask, but please do not hesitate to reach us for those who are here to join or who want links to some of these resources that I've shared, or anything you have that you'd like us to support or to respond to. Please do not hesitate to write us at africa@pine.bao, africa@pine.bao, and you can equally try to chat us up on our social media pages. We would respond to you. So on that note, I'd like to end this session today, except where there is any questions or comments quickly. Do we have any? So great. It's been a wonderful time this evening. We appreciate everyone who was able to join us. So we meet again next week, Wednesday. However, the training continues tomorrow. It continues tomorrow, Friday, and all along until we meet during another live session next week. So for paid participants or those who still want to join, the training has not stopped. The live session will only return next week. The training continues. Take advantage of the materials and the resources on the platform so as to get the very best of the premium access you have. Okay, I think that is what I would say, because after 6 weeks, for those who are even paid participants, those premium courses will return to being premium, even for you, meaning you won't be able to access them. So while the time lasts, please kindly, do the very best, maximizing all of these premium courses. And then a good night, everyone. See you on the Slack channel and on other platforms. Thank you.